Hello and welcome. Today I want to talk about solid tumors versus liquid tumors, more specifically how this categorization distinguishes between the two, what differences differentiate a solid cancer or a solid tumor from a liquid tumor, and also how do the different cells behave? Are there alterations to the cell behavior in a solid tumor versus in a liquid tumor? Because in the end, if the cells do behave differently, that might have an effect on how we can treat these cancers, on what options are available. And in the end, this is the final aim of understanding cancer, is to enable better treatments and thus better cancer patient outcomes. Now, both solid and liquid tumors are cancers, so they share the fundamental property or fundamental hallmark of cancers of uncontrolled cell growth, of uncontrolled proliferation, and thus the creation of more cells than is necessary. They also share a subsequent accumulation of these cells. This is also a part of what makes cancer dangerous, isn't just the uncontrolled growth, but it is the fact that the cells don't die or they aren't cleared out of the body afterwards. Because if that was the case, then you wouldn't have abnormal dysfunctional accumulation of cells. Now, like I said, this is something that both liquid tumors and solid tumors share since they're cancers. Where they differ is in the mode of accumulation. So where within the body do the cancer cells, do these superabundant cells accumulate? How do they behave? And how does that affect the function in your body? And of course, how does that affect treatment? Now, if you don't know who I am Cedric. I have a bachelor's degree in molecular biotechnology, and together with my brother, I am one of the co-founders of Marchwood, where our aim is to help cancer patients outlive their disease, outlive their prognosis, by informing them about recent scientific findings and what those scientific findings mean for potential avenues of treating cancer. We also don't just want to create a cult, so you can basically read up on anything that we say. You can read up on our understanding and on our claims of how you can treat cancer online by going to marchwood.com/mosaic. It's a living project that we have there. It's a book that we are currently in the process of writing, where we've written down completely our understanding of cancer, what the implications are for the treatment of cancer, but also because we want to inform people and not create a cult around us where people just blindly follow us. You can find all the scientific sources. So all of the claims that we make have scientific sources that allow you to double check us, to go into these scientific sources on your own and to better inform yourself so that you don't end up making decisions because you become a cult follower of some people on the internet. Because let's be honest, you shouldn't just blindly trust us. You should double check us and you should be skeptical of anything we have to say. We might be snakes oil salesmen for all you know. Now, of course, as we go on, we hope to earn your trust, but we hope to earn that trust by allowing you to double check us again and again. So, like I said, if you are curious to learn more about our understanding of cancer and why we have our understanding and our methods, you can go to marchwood.com/mosaic to double check us and to read up on us. Now, let's get into the meat of this video. Let's get into the differences between solid and liquid tumors. Like I said in the beginning, there's some similarities in the uncontrolled growth and the fact that cells accumulate, but they differ in where they accumulate. Now, to look at this more closely, I think most people will be aware with solid tumors. These are the majority of tumors in adults. They make up around 90% of the tumors that we see. And I think this is one of the reasons why people, when they hear a tumor, will instinctively think of a solid tumor. Now, this is the stereotypical tumor that forms a cohesive mass somewhere in your body, maybe in the heart, maybe in the brain, prostate. All of these tumors form a cohesive mass where the cells are attached to one another. It's kind of a lump, right? And this is why solid tumors, sometimes when they are not too far advanced, can be surgically removed because they are kind of localized. They are localized in a certain site in your body, and they are cohesive, which means we can cut out around the cancer cells, around this lump of cancers, and we can remove them. And if that is successful, of course, if all the cancer cells are in fact removed by a surgical procedure, that can completely heal the cancer. Now, as you might also know, solid cancers can metastasize. This is where cancers or solid cancers spread from one side to another in your body. So they might spread from your liver or from your muscles, from your skin into your brain or into your lungs, into your bones even. And in such a case, of course, surgical procedures are no longer possible. But still, even in the case of metastases, the newly formed solid tumors are themselves cohesive masses. So they might jump over to another spot, another site in your body, but they won't, I guess, liquidize is a way to say it, which puts us, or which transitions us over to the liquid tumors. Now, these, as you might have guessed by now, they don't accumulate in a solid context, but rather they spread throughout the circulatory system in your body and kind of travel around in these liquids, and they are not cohesive solids and called liquid cancers or liquid tumors. Now, these are most commonly leukemias or lymphomas, where the blood cells are overabundant and they end up filling up the blood with too many, for example, in polycythemia vera, filling up the blood with too many red blood cells, which then means that other cells, the blood becomes too thick, other cells are kind of outcompeted, and the function of the blood is inhibited. Whereas with solid cancers, or solid tumors, these are often killed by displacing other organs. So, for example, a tumor in your brain might create too much pressure, it might grow too quickly and destroy the tissue around it, which, of course, having destroyed brain tissue isn't great. So these are the differences kind of in the context in which liquid tumors and solid tumors arise. Now, we know in general that cells are extremely context dependent. I mean, after all the cells in your body have the same genome. However, you might also realize that you have completely different cell types. You have skin cells, you have neurons, you have muscle cells, you have intestinal cells, you have cells for seeing light in your eyes, you have olfactory cells, you have secretory cells, and so on and so forth. This is because the cells and their behavior is extremely dependent on the regulation around them, on the environment. They might get different signals, they might have different nutrients available, they might receive different stimuli, such as stimuli from light, and all of this causes the cells to change their behavior based on the surroundings. In fact, it's even possible to take muscle cells from a human and de-differentiate them, is what it's called, to basically create cells from these finely differentiated muscle cells, and then redifferentiate them into different cells here. So you can take human muscle cells and you can kind of backtrack and create neurons from them. And this is only possible because the cells are so dependent on their environment and on external context for regulating their behavior. And of course, cancer cells are no exception. Cancer cells don't exist in a vacuum. They are also extremely dependent on the cells surrounding them. And they will even create something called the tumor microenvironment, or TME for short, which is basically a sub-environment that they manipulate. So you might have a lump of cancerous tumor cells right here. All of these are small tumor cells, and they will manipulate this environment by acidification or potentially by altering the metabolism. Or, for example, creating new blood vessels, or they might even recruit immune cells, which normally are intended to kill and hunt the cancer cells, and kind of corrupt them in a simple way. Of course, it's more complicated than that, but they kind of corrupt them and recruit them to their own purposes. So this is kind of things that the cancer cells do to manipulate their environment to make it more. Let me just finish writing this out. This is something cancer cells may do to make the environment more suitable for their own growth. So basically, they want to enable themselves to outcompete other cells and to give themselves the best advantage that they have. So as we can see here, cancer cells don't exist in a vacuum, and the environment may very well affect growth. And given that we just in the beginning talked about how different the environment between solid tumors and liquid tumors are, the question is whether the vastly different environment could not also affect the cancer cell behavior. I mean, that is a very sensible it's a very sensible suggestion, given that we know that cells are very dependent on their context, and that the context in which the liquid tumors and solid tumors find themselves are very different. And there is certainly a truth to that. However, the solid tumors and liquid tumors, especially in their growth, might be more similar than, or are much more similar than one might expect. Because despite the liquid cancer cells, the liquid tumors accumulating inside of circulatory system, the growth actually occurs under similar conditions. Now, this might be a bit counterintuitive at first, but if we want to understand that, we can look at the growth in solid tumors and then kind of compare that to how liquid tumors grow. So if we have solid tumors, these are probably easier to understand for most people compared to the liquid tumors. We have kind of normal cell growth. So we have a cell here, this is a cancer cell, and it will split up into two daughter cells, two descendants, I will call them. And these two descendants are, for all intents and purposes, the exact same as the precursors. So you have a cancer cell that gives rise to two descendants, but these descendants are themselves, of course, cancer cells. And they can then also, once again, divide and give rise to new cancer cells, which once again will be somewhat similar to the precursors, of course, save for mutations, and they will then be able to divide again. And I think you see where this is going. This is going to give rise to exponential growth, because as the new cells are created, which are effectively clones of the precursors, these subsequent descendants are able to divide again, give rise to new descendants, which are themselves again able to divide. So this is kind of a symmetrical, non-hierarchical division process. Each cancer cell divides into two new cancer cells that are effectively the same, and as the cancer cells expand, the productive capacity in quotation marks goes up, because of course if you have two cancer cells that can produce new cancer cells, that's going to go a lot faster than if you have one cancer cell. And then after one round you have two, after the next you have four, after the next you have 8, and then you have 16, and so on and so forth. Now, liquid tumors are somewhat different in this regard, but interestingly enough, they will also proliferate in a solid context. So despite the tumors themselves accumulating inside of the circulatory system, inside of the lymph nodes or inside of the blood, the cancer or the growth itself will happen in a solid context. Now, it will differ somewhat. So this is solid, and in liquid tumors, the growth actually occurs asymmetrically. So where with the solid tumors, we saw this even split from a precursor cell into a descendant cell, and they were basically the same. With liquid tumors, you have these precursor cells, but they don't divide into the same cell types. So they divide into another cell that is basically a clone. So it is much the same, and it can still proliferate itself. So this can go on proliferating. However, the other cell that is created in this process of division with liquid cells, it will not be a clone. So it might look different. Now this is definitely not how the cell, or this is a schematic. I'm not saying this is exactly how the cells look, but these cells are differentiated and they are unable to produce new cells, and they will then enter circulation. So these go into circulation, the descendants. One second. Ooh. So these differentiated descendants will not be able to divide themselves, and they will enter circulation, and this is where they accumulate. But that means that for solid tumors, we have exponential growth and a accumulation inside of the solid cohesive mass as more and more cancer cells grow and remain in the same spot. In comparison, you have the liquid tumors, which are linear growth, because the new cells that are created, only one of these, if we go back here, only one of these is capable to produce new cells. So after one cycle, you have one cell that can create two new cells. But after the second cycle, you still only have one cell. And after the third cycle, you still only have one cell. So the productive capacity, so to say, which I said expanded up here, if you have one, next round you have two, then in the next round you have 4, and then in the next round you have 8, right? You can see that this number of cells that can divide, it increases. It doesn't with the liquid tumors. So the productive capacity remains the same, which means that every round that cells split, you have the same number of cells that are dividing, and then you have the same number of cells that are created which leave into circulation. So this leads to linear growth. And of course, it leads to accumulation of the cells inside of the blood, because it is a one-way street. The cells will leave from the maturation organs, from these solid niches where growth occurs, into the bloodstream. And then the next cycle occurs and more cells are released into the bloodstream. And this is why liquid tumors accumulate in the bloodstream, but actually grow in a solid context. So that actually means that despite the accumulation being in different places, one in a solid kind of lump somewhere in your body and the other throughout the circulatory system, the growth conditions are actually extremely similar. Now, what's also interesting is that solid tumors sometimes do transition into the circulatory system, which is the case in metastases. So if you look at metastases, we can see that sometimes you have a lump here somewhere, maybe, let me draw it a bit more. You have a human, and he currently has a cancerous lump in his leg. This cancerous lump in his leg then or some cells from this might transition into his bloodstream. So they might go over here into his veins, right? And then they kind of travel along, and at some point they might think to leave the bloodstream again, and they will settle in the next tissue. So they might leave and settle back here inside of his arm or inside of his brain or inside of his lung. Now, during this process of migrating throughout the body, the cancer cells, the metastatic cancer cells, which we call the cancer cells which migrate through the body to create metastases, they actually don't divide. And that's interesting because normally cancer cells, especially metastatic cancer cells in a solid context, are highly proliferative. So for some reason, when the cancer cells from a solid cancer go from a transition from solid context to liquid context, they at the same time lose the ability to proliferate. So they become unable to create new cells in this liquid context until they settle again. Once they settle, so once this once this occurs and they've settled in the new organ, they become capable of creating a new cohesive mass in a solid context and they start growing again. But this is eerily similar to how liquid tumors in their liquid context are not able to grow. Now, of course, a little caveat here, this is biology, it isn't 100%. So sometimes metastatic cancer cells, whilst migrating, or liquid cancer cells, whilst in the liquid tumor, whilst in the liquid state, do maintain a ability to proliferate to a certain extent. However, this proliferative ability is still significantly reduced. So if you have a patient who has a liquid tumor, where his liquid tumor cells do retain the ability to reproduce, to proliferate, the majority of the liquid tumor cells will still be from the proliferation within the solid niche, much like the metastatic cancers, which might be able to divide slowly, however, not to an extent that would be significant and would impact its health. And that means that if we can somehow stop growth inside of these solid growth niches, so if we can stop growth inside a solid context, then we can completely get rid of the symptoms from cancer, because both liquid cancers and non-liquid cancers and solid cancers both grow in a solid context. So it doesn't really matter whether it's a solid cancer or a liquid cancer for our options to treat them. It's because the conditions inside of these growth niches are so very, very similar. And that means that even though a liquid cancer and a solid cancer might at first appear very different, because of the different accumulation sites, because of the different effects on the system, where liquid cancers are more impactful on the blood, whereas normal cancers are more impactful on the organs throughout the body, on the cohesive structural organs. Both cancers are proliferated or are grown under the same conditions, and that means that if we can stop growth under these conditions for the cancer cells, we can stop the creation of, or the growth of, these cancer lumps, or we can stop the creation of new cells that enter the circulation in the body, and then we don't have symptoms in either case. Now, I think diving into the exact ways in which we can stop this growth here would go a bit beyond the scope of this video, because I wanted to focus on the differences between solid tumors and liquid tumors, because it is something that appears very different at a first glance, but once you look deeper, you actually see that the liquid tumors are basically proliferating like solid tumors under the same conditions as solid tumors, and simply are ejecting their cells into the circulation or into the circulatory system. Now, of course, liquid tumors, like I said, they have linear growth rates, which means that they are not as rapidly explosive as normal tumors can be. However, the mode and the conditions, that means, for example, acidic tumor microenvironment, hypoxia, abnormal mitochondria, high reliance on glucose, all of these factors are shared between the solid tumors and the liquid tumors. So if one takes a normal tumor cell from a solid tumor and a tumor from a liquid tumor that we had before, if we analyze these cells, these non-proliferative cells that enter circulation, so these that go into circulation, they might very well be somewhat different from normal cancer cells. But we also have to remember that these aren't the cells which are proliferating. If we instead focus on the cells which do the proliferation, we see that they share the same hallmarks. They share the same metabolic abnormalities, they share the same mitochondrial damage, they share mitochondrial fission, they share genetic mutations, they share the microenvironment being hypoxic, being acidified, and because of all of this, the potential remedies which are based on exactly this state and this environment around cancers and the state of cancer cells themselves, which are proliferative, can be applied both to liquid tumors and solid tumors, regardless of whether they stem from research based on solid tumors. For example, one of the main, I would say, inspirations for this video was when I was researching how alkalization, which I was aware of from solid tumors, might be applied to liquid tumors, because my initial reaction or my initial thought was that because solid tumors have their own specific microenvironment, whereas liquid tumors circulate throughout the blood, that alkalization might not be applicable to liquid tumors, since it is much harder to completely alter or change the composition of the blood, because it's very tightly regulated. However, like I said here, the growth occurs under similar conditions, and because of that, it actually ends up being that despite alkalization of the liquid context perhaps being difficult, alkalization of the liquid context isn't necessary, but rather alkalization of the solid context is necessary and sufficient to hinder and potentially even completely stop proliferation. Of course, I think this is kind of where I want to draw the line. If you want to look into how these other remedies that I've just talked about, that I've kind of touched on, where they stem from, what's the scientific basis, what's the cellular basis behind these other remedies, what was I talking about with mitochondria, what is this alkalization? There are other videos on our channel. I will link a video to alkalization and to the mitochondria themselves, how they are fundamental to cancer and how they can both be used as attack points against cancer in the description. But of course, there are also many other aspects of cancers, of their growth conditions. For example, what I talked about with hypoxia, or with reliance on glycolysis, which you will find videos on our channel. Or if you like reading, you can also go over to marchwort.com, where we publish articles on exactly these topics. But I think that is all I had to cover for this video. And I hope you learned something. I hope that this was interesting to you, and I hope that it was presented in an understandable manner. And I wish you a great day and swift healing and lasting health. Peace out.